Um, Good. Good. In a normal year, you would have had now the opportunity to learn how to dissect larvae and to do nice chromosome preps. Unfortunately, we have to skip that and we continue with the second genetics lecture. Einführung in die Drosophila-Genetik, moderne Methoden, reverse genetics. So, reverse genetics versus forward genetics. So reverse genetics, as I said, starts from the sequenced genome, and you say, okay, this gene here has homology to another gene in human, for example, and you say, I would like to know what the effect of mutations in this gene in the fly are. No mutations are known. That's the starting point. So you decide, for example, I choose gene CG 5204 because this is could be very interesting. And then you use the reverse genetics tools, which we are going to talk about in a minute. And then using these tools, you can make gain of function or loss of function mutations. And once you have these, you can start the genetic characterization of this gene CG 5404. 204, yes. The problem with forward genetics is you can not mutate every gene to give a phenotype. That is not the case. Many genes, when mutated, do not give you a phenotype. So when you go the reverse genetic approach, if you go down that path, then of course it's also possible that when you mutate it using the reverse genetic tools, that you will not have a phenotype. It's not an essential gene, in other words. Or the effects are so mild that you will not necessarily have a clear-cut phenotype. So that's always possible, even in the fly, even more so in organisms which have larger genomes where you have multiple copies of the same gene doing separate things. Might be that by far not every mutation in the gene, even if it's a null allele, will give you a phenotype. The fish people in our lab know what I'm talking about. Okay, what do we have to learn if we want to use reverse genetic tools? What are these? What is the basis for it? Transgenic flies. You have to be able to make transgenic flies. If you cannot make transgene for your organism of choice, then this is a major drawback, and you will see that essentially all or almost all of the reverse genetic tools are based on the production of transgenic flies. And of course, we are now in 2020. The other thing is CRISPR. I'm sure everybody has heard extensively now about CRISPR, so I will only briefly mention it, but I think Marcus will also talk about it on Wednesday morning. But a few years ago, nobody knew what CRISPR was, so everybody knows now what CRISPR is, and that's good. So CRISPR is very important for us because it gives us a tool to introduce mutations relatively easily and quite fast. Okay, transgenes. What is a transgene? I have shown you for the white gene that many transposons have integrated into the white gene and cause the phenotype, leading to different eye color phenotypes. And the transposons, they have been discovered by this woman here, Barbara McClintock. She worked on corn and was studying these variations you see here on the picture on the right. So she studied this phenomenon and realized or proposed that these are mobile genetic elements which are able to move around in the genome. And she did her work in the early 1950s. Of course, nobody believed her. How should there be anything like a mobile genetic element? But she was right, and she received the Nobel Prize in 1983, already at quite high age. Okay, don't be discouraged if you have a important to you discovery. Not everybody might believe what you want to let them know, but at one point people might realize that you were actually right. So, but of course, not everybody makes these kind of important discoveries these days. Anyway, she discovered the mobile genetic elements, and I again show you this slide here for the white gene. There are many different mobile genetic elements in the fly, and the one which was studied around 1980 was the P element. You can see there is a P element here in white, there is a P element here in white. Yes, these are the only ones I can see at the moment. Anyway, one of these two was cloned. You should be aware that the molecular cloning techniques were developed in the 1970s, and it was before impossible to do this. So around 1980, people started to look at these mutations in white and found that these two here had P element insertions in white. I'm sorry. So they could sequence this DNA once they had cloned it. The people who did it were actually these two here, Alan Spradling and Jerry Rubin. Jerry Rubin is again the same person who was very important when 20 years later, when the Drosophila, the fly genome was sequenced. So we will spend now a little bit of time to look how the P element looks like and how it can be used, how it was engineered to make it a tool to make transgenic fly lines. So this is a P element, the transposon. It's short, it's only 2.9 kb in size, and it contains one gene, this blue gene here. And it also contains these funny ends, inverted repeats they are, or ends containing inverted repeats. So these are the two important components of the transposon. The gene in here codes an enzyme called transposase. It's required to let the transposon jump around in the genome, and the P element ends, they contain 31 base pair terminal repeats required to jump around. So as long as a P element has this transposase and the P element ends, the transposon can move around in the genome. When people were sequencing other p elements they had isolated in the genome, they found that there were also what they called non-autonomous p elements. Here, this is the complete p element. It's autonomous. It doesn't need another p element to move around, but there are also non-autonomous p elements. They have internal deletions which destroy the transposase. One could also imagine that there are p elements which have deletions in the terminal region that would prevent them from jumping around, but they would still produce transposase. That's also a possibility. But anyway, they found there are autonomous and non-autonomous p elements. The non-autonomous p elements, as they are shown here, they require that if they want to move around in the genome, they require the presence of an autonomous p element which produces transposase. And if there is transposase, these p elements here can also move around. But they cannot move around if there is no autonomous p element around in the genome. So this is a very important result, observation. There are two different types of transposons. Then people realized that there are different types of Drosophila lines. There were so-called M lines. They do not contain p elements, autonomous p elements in their genome. And there are so-called P lines. These contain many p elements in their genome. The number of these p elements in the genome is variable from stock to stock. And the important thing is we are happy that we still have these M strains, because if you look nowadays in the wild-type populations, essentially all of them are P strains. But at the time when people started to work with Drosophila 100 years ago, they also had still M strains. And that is very important because you can only make transgenic fly lines if you have an M strain, with other words, which do not contain autonomous p elements in their genome. And you will realize soon why this is very important. Okay. Again, the two important components of the p element, the inverted repeats, which are required for the integration or also for the excision out of the genome, and the transposase gene makes a protein and interacts with these inverted repeats here and catalyzes integration or excision of the transposon. That's very important. So people said, this is very interesting. If we separate these two components onto two different plasmids, we can control the insertion of a transposon much better than if we have a plasmid which contains both of the components. So they designed two vectors. A p element vector, which is basically a normal plasmid, as you have learned it in microbiology, when you were transforming bacteria. So a p element vector and a helper p element. This helper p element is also basically a simple plasmid. So you have these two different plasmids. The p element vector, it contains the inverted repeats, but not the transposase. The helper p element doesn't contain inverted repeats, but it contains the transposase. So with other words, the p element, if it somehow receives transposase, it can integrate into the genome. The helper p element, since it doesn't have the inverted repeats, it will never integrate into the genome. If you bring the helper p element into the embryo, it is present there, but it will be diluted out and it will be lost. So, as you will see, the p element vector and the helper p element here, they are brought together into the fly embryo, where the helper p element produces transposase and causes, with relatively low frequency, this p element vector to insert into the genome. That is very nice because you don't want that this event happens too often, because you want, in principle, one p element vector to insert into the genome and not multiple. You want a single one of them there. The helper p element will be lost, transposase is no longer around, and the p element vector cannot move around anymore. So this is the dual system which has been developed, consisting of a p element vector and the helper p element. So the p element vector, how does it look? It's a circular plasmid. I have drawn it here as a linear, but it's in print. It's really a plasmid, a circular plasmid, and you need to be able to work with it in bacteria. That's why it has an ampicillin resistance gene, for example. Kanamycin is also possible, and an origin of replication. So these green parts here, you need to manipulate the p element vector in bacteria, because you want to clone your DNA of interest in here, the multiple cloning site. Here you will insert, by using restriction enzymes, you will insert your DNA of interest. In addition, you need a means to identify your transgene once it has inserted into the genome. And for that reason, this red block here is present, the white gene. You have heard about white before. I have told you that its gene product is required to import pigmentation, also pigment, into the eye and give eye color. So this here is our transposition marker. It will help us to identify an insert. I show you how this looks in a moment. Then the important thing, what we need, are these p element ends, the 3' p element end, the 5' p element end, and the 3' p element end. This is the site where the transposase will interact with and mediate the insertion into the genome. So that is basically how this p element vector looks like. Very simple. You can now, as I said, introduce your DNA of interest in here, bring it back into the fly, and study what effect this DNA of interest has in the fly. That's how you make them. I told you that our transposition marker is white. Here, white. It's a defective p element in a way because it doesn't produce transposase. It's not non-autonomous, in other words. Here we have the p element, the helper p element, which makes transposase, but it cannot insert because it doesn't have the p element ends. So you have a fly stock which has white eyes, white minus, we call it. It's null for the white, it doesn't produce eye pigment, the eyes remain white. Now, using a very fine micro capillary, so a needle basically, in which you fill in these two plasmids, we inject an embryo. This is the embryo. This is the anterior end. This is the micropyle, marking the anterior end, and the posterior end. In the fly, in the posterior end, the germ cells will develop. About 90 minutes after embryonic development has started, the germ cells are set apart, and of course we want the germ cells to have an integration of our P element. So we have to inject early into young embryos. We inject these two plasmids, and if we are lucky, the transposase will catalyze the insertion of this plasmid. We let these embryos develop to adults, here, G0 generation, and we cannot identify yet whether the P element has inserted or not, because white is a cell autonomous gene, meaning that it only can affect the cell in which it is present. And since the eyes develop on this side, none of the cells which will later develop into eye will obtain this plasmid here. That's why these flies here still have white eyes. When white is expressed in the germ cells, it has no effect on eye color. So what do we have to do? We have to cross these flies with each other. They have white eyes. And now in the next generation, since they might be transgenic in their germline, the progeny, once in a while, will have not white eyes like these all have white eyes, but it's transformed. It has obtained this vector here, which we have injected into young embryos. Now, in this generation, in the G1 generation, we can spot the transgenic flies and breed them. How do these flies look like? That's shown here on this slide. So these are the flies into which we have injected our P element vector, has white eyes. Then we do what I just showed you. We bring back the DNA into the germline. We cross the resulting adults, and we get eye color back. You can see here I have drawn the homologous chromosome. There is now one insertion here which causes a clearly pigmented eye, which can be easily separated from this type of flies here. If you see a fly like that, you have your transgene insert. If you have two copies of the same, after you have crossed them, of course, you make a homozygous stock, and you can see that the eye pigmentation is clearly darker than the heterozygous insert, and this indicates to you that the white gene is dosage dependent. You get darker eye color if there are two copies of the white gene. On the next slide, I show you that depending on where the P element inserts, you can get different eye colors. So this is again white, no insertion. You can insert somewhere where the white gene is barely expressed. You get weak yellow, pale yellow, or you get yellow eyes, you get dark yellow eyes, you get orange eyes, dark orange, brown, red, red. So depending on where the P element is inserted in the genome, the expression of the mini-white of the white gene will be different. This also reflects the fact that the euchromatin, where most of the transgenes are inserted, is not everywhere the same. And we refer to that as position effect. Depending on the position on the chromosome, the white gene experiences a different effect. Either it's more active or less active, and that we refer to as position effect. Normally, the expression of white is uniform over the whole eye. In rare occasions, you can get quite funny looking patterns of white, and but this is rather rare, but also has been obtained. Okay, that's how you make transgenic flies in a nutshell. Now, if you have isolated one fly, you have to turn it into a stock with which you want to work. So here, let's assume our P element marked with white has inserted on the third chromosome. On the X chromosome, it's white. It's white minus. Otherwise, if it would be white plus, we would not see it. So on the X chromosome, we have a white minus background, we call it. And we could identify this insert because the white gene here inserted on the third chromosome complements for the loss of white on the X. So we have such a male, a single one. This one would die at the latest after 2 to 3 months, and we would have lost it. That's, of course, not what we want. We want to be able to make a stock which will be there forever, more or less, until you are fed up and you want to discard it. That's also possible. But anyway, we want to make a stock with which we can work and which will be there for as long as we want. For that, we cross this male here with females, which we take from a stock, which we are also keeping. It is white minus on the X. That's important. It has a mutation, a lethal mutation on the third, balanced over TM3's double. So blue is the balancer chromosome. And the idea is to make a stock where this chromosome, the red chromosome, is present and the blue chromosome is present. If you cross these, according to Mendelian genetics, you get these kind of genotypes. Here, blue, red, yellow, blue, blue, yellow, yellow, yellow. So I told you it's important that we bring these two chromosomes together. How can we identify that they are in the same fly? So we are still in a white minus background because both parents have white minus on the X. Now, if we look at the phenotypes, these here have eye color because of the red chromosome, and they are stubble. Stubble indicates balancer is there. These flies here, they are white minus because they don't have the red chromosome, but they are stubble. These guys here, this genotype, they have eye color, but they are not stubble because they have received this chromosome from the mother. And we have the guys which are only white minus. So no insert on the third and also no balancer. So based on the phenotypes, we can now select those flies which are interesting to us. And these are, of course, these here, because they have now the red and the blue chromosome in the same fly. We cross males and females with each other, and we have established a balanced system. The only viable offspring are the homozygous flies or the heterozygous balanced over TM3. If you cross these again, you again will get the very same flies. So with other words, we have a balanced stock. And if you want, you can make a homozygous stock as long as this insert here is homozygous viable and fertile. So this already indicates to you not every insertion of a P element causes a mutant phenotype, and that is very good because we would not like that. It's better if P elements end up in a place where they do not have a mutagenic effect. That's a very nice thing, even though many of the P elements insert actually in the 5' ends of gene, most of them do not cause a phenotype. Many of them do not cause a phenotype. So this slide here, we will not go through right now. It's part of the genetics questions. You will deal with that in the genetics questions, but this is basically the blueprint to answer one of the questions there. I will not go into it. We will discuss about it at length when we do it on Wednesday or Friday. Okay. This is how you make transgenic flies. Very important. Over the year, probably 10,000 of transgenic constructs have been injected, if you look in the whole fly field, and many of these lines are available. If you ask this lab kindly, they might send it to you. Many of them are stored in stock centers where you can get them. Very nice and good, but there are now other systems which we have to introduce to you to understand the experiments you are doing in silico, basically, in the next few days. I'm sure you have heard about the Cre-Lox system. Cre is a recombinase. Its target site is LOXP. And if you have two LOXP sites somewhere in the genome relatively close to each other, you can easily make a deletion by introducing the Cre recombinase. There will be a recombination event between the two LOXP sites and you will lose the DNA. I'm sure you have heard that in lectures dealing with mouse genetics. It's also reversible. So you can use the LOXP site again to do another round of Cre treatment. And as I said, it has been used to make in mouse to make deletions and duplications. In the fly field, we'll use much more frequent the flip-base FRT recombination system. The enzyme analogous to Cre is called flip-base. The target site on the genome is called FRT, flip-base recognition target. And as with LOXP, the FRT remains an FRT once you have done a recombination event. That's also very important. In the fly, we use it like Cre in mouse, we use it to make deletions and duplications. And the very important application in fly is mitotic clones. So if we want to make mitotic clones, we use the flip FRT system, and Gustavo will tell you more about it tomorrow. Then we have the integrase system. This is about in use since about 5 to 10 years. It has been introduced in the fly field. Like these systems here, they have been adapted from prokaryotes. So here the enzyme is called integrase, and you have its target sites like analogous to LOXP and FRT are called at P and at B. And once you make a recombination between at B and at P, then these sites are no longer usable. They then are called at R or at L, and they are not reversible. So we use them to make transgene insertions. I will introduce to you now in more detail the flip-base and the integrase system. I mentioned that if you use p-element for insertion to produce a transgenic insertion in the fly, the p-element will insert at random. You cannot control where it will insert. This is pretty much okay, but sometimes you would prefer a situation where your transgenes are always inserting at the same insertion site, because you don't want your different constructs to experience different position effects. If you want to go in that direction, you first make a transgene, a p-element transgene here, the p-element ends, the marker, and you introduce in this way, so in the cloning site, you have introduced an at-p site. You make a transgene, as I told you before. This is called integrase docking site. And you can map it, and you know it's there in the genome. That's where I want to insert all other constructs you have been making. And these are the constructs you have been making. They contain a second marker, your DNA of interest, which you want to study, and the at-B recombination site. Then you provide integrase, and there will be a recombination between at-B and at-P. You can isolate this insertion because beside marker 1, you can also identify marker 2. And if you look then by sequencing and so on at your insert, you will realize that now this site here also contains your DNA that you have injected. Your DNA of interest is there, and you identify it through the presence of marker number 2. So that's how we use these days often the integrase system rather than the p-element insertion system to make new transgenic insertions in fly. Both are of use, but we use these days quite often this one here. There is a second possibility to use at-P system. There you have, beside marker 1, you have 2 p-elements here, 2 at-P sites, I'm sorry, but they are present on a p-element. These are the p-element ends. And the plasmid that you inject contains 2 at-B sites in the same orientation as the at-P sites in the genome. Now, instead of inserting the whole plasmid, as in the example before, you are only inserting the DNA located between at-B and at-B here. In this way, you lose marker 1 and you identify your flies which have successfully done this integrase insertion by looking for the loss of marker 1 and gain of marker 2. Again, through the recombination at L and at R are formed, and since they are no longer a substrate for the integrase, you lose, they cannot be, you cannot lose the DNA which is inserted here, even if integrase is present. Okay. That's so much for the integrase system. Now I will show you a few more slides, and then we have a little break before we move on to the last part of this lecture. So how does the FRT work like? Or locks would work in a different, in a very analogous manner. For example, if you want to make a deletion between two FRT sites, so the blue arrows here are FRTs, you provide, they are in tandem, you provide the flip base, and as a result, you lose the DNA in between the FRTs, and this part here is lost. Your chromosome now has a deletion in between the two FRT sites. If they have different orientation here, one in this orientation, one in that orientation, the FRT, if you then do a recombination here, you will get a inversion. The DNA is not lost, but what was here is now there, you see? And what was here is now there. So you really have to be well informed about the orientation of your FRTs. Depending on what you do, this is very important. This deletion here, this deletion approach, has been used by the Fly field to make deletions in the whole genome. That was done in the following way. First of all, hundreds of p-element insertions were generated which contain an FRT. They were mapped, and then they were crossed together in the presence of flip base. And here, in such a case, you have this chromosome comes from one stock. The purple chromosome comes from a different stock, which has an insertion here. This has an insertion there. If you bring in flip base, you will now delete everything between this and this FRT, causing a deletion here of region B. As a byproduct, you also get the duplication. Here, there you have a duplication of B. That's sometimes also useful, but this here has been of most interest, and in the Fly field, as I said, hundreds of such deletions have been made in such a way. For example, here we have deficiency 2L-ED 2809 was created between FRT containing element 1 and FRT containing element 2. The first one maps here at position 76,365. The other one maps here at 72,671. This is on 2L. And if you make the deletion, you have a 5,306 base pair deletion, which could be of interest to you because your gene of interest is located there. And in that way, hundreds have been made. You can see that some of them are quite large, half 500,000 base pairs almost. But if you want, you can also only delete a few base pairs if element 1 and element 2 are relatively close to each other. Then what happens in the case before, we had the two FRTs were in the same direction. What happens if they are not? Flip base will anyway mediate recombination between the two FRTs. The chromosome has to kind of turn around a little bit so that the FRTs are aligned in the same direction. And as a consequence, you get a chromosome here, A over to B, a dicentric chromosomes. and an acentric chromosome. So this is, of course, again, the same problem as we were discussing in the previous lecture for recombination events in the inversions, causing acentric and dicentric chromosomes. These recombination products are cell lethal and you can never obtain them. Okay, I think we have now a break until about 20 past, I would say. And we meet again in 14 or so minutes. And I will introduce you then the last couple of slides of this lecture. Okay, so see you in a minute or 10 minutes, 15 minutes.